Hi everyone, welcome back after spring break. This is definitely a little bit of a different format than any of us expected, but hopefully we can make this work as well as we need it to for us to complete the semester. If you haven't already read the Blackboard announcement that was also emailed to you that was sent out on Sunday, please make sure that you read it. It has a lot of information on how we are going to be approaching things for this week and while we are not meeting face-to-face. So today my goals are pretty simple. I want to just kind of briefly introduce Module 3, Human Genetic Diseases, and try to give you the information you need to make an informed choice of a human genetic disease for your research paper. Remember that the topics are subject to approval just to make sure that they will work well, given our rubric that is posted on Blackboard. And we need to make sure that each student has their own topic. So there's a running list of selected topics that's posted on Blackboard. And reminder that you'll need to let me know if you haven't already your topic of choice by 11 a.m. this Thursday, March 19th. Okay, so what do you want to consider when you're picking a topic or when you're just trying to understand human genetic diseases? Well, there's a good resource posted at the top of this slide, which is a free textbook, essentially, that the journal Nature has put out. So you can click on that link to read a quick overview about genes and disease. And you can also learn more about simple or monogenic diseases versus complex diseases. Okay, so human disease categories include monogenic, which just means they're caused by a single gene, that each person who experiences this disease or disorder, all of those individuals have a mutation in the same gene. Another category would be chromosomal mutations, and a third category is sometimes called multifactorial or complex diseases. And their name suggests they are not caused by just a single gene. Sometimes it's multiple different genes that can give rise to the same disease or disorder, or interactions with multiple genes or the environment give rise to this disease. So I'm going to tell you just a little bit about monogenic versus multifactorial diseases, and then we'll spend a tiny bit of time talking about how to use OMIM, a really helpful online resource. Okay, so first, these single gene or monogenic disorders. There's a nice list of them in Table 1 of that online reading. Muscular dystrophy, cystic fibrosis are examples of autosomal recessive disorders, where again, there's just a single gene that is mutated that gives rise to those diseases. Autosomal dominant disorders would include Huntington's disease, myotonic dystrophy, hypercholesterolemia. There are also X-linked diseases like hemophilia. So and that would be an X-linked recessive disease, and there are some X-linked dominant diseases as well. One of the considerations that you will be writing about, or one of the considerations for you, and one of the topics you'll write about in your papers, is the inheritance of your selected disease. So the inheritance patterns for these monogenic diseases are generally simpler than the complex diseases. For these diseases, you will see published pedigrees and pedigree analyses of large families, and we will be looking at some examples of these in the coming days. They generally have typical Mendelian patterns like you've learned about in intro genetics. Right now, as of February 2020, we know of 6,604 phenotypes or human genetic diseases that have a known molecular basis. So there are a lot of possible topics, not just that list I posted on the prior slide. And at the same time, there are still many phenotypes and diseases for which the gene is not yet known, and those will not work well for your topic for your research paper. Okay, so in contrast to the simple or monogenic diseases, complex diseases are, as their name suggests, more complicated. However, they're also more common. And as you consider what your topic will be, you may have a personal connection. You may know someone with a particular disease and you'd like to learn more about it. So these complex diseases are more common, and this might be a reason you would choose one of these diseases: Alzheimer's, asthma, Parkinson's, MS, some of the autoimmune diseases. What makes them complex is that they don't follow typical Mendelian patterns in these pedigrees. They're not typically recessive or dominant. Instead, they're caused not, again, by a single mutation in a gene, but a combination of genetics, environment, and lifestyle. Okay. So we don't see those simple patterns, and the pedigrees can look quite complicated for these reasons. There can be incomplete penetrance, which means that individuals that have the same genotype don't all show the same phenotype. Similarly, there can be variable expressivity, where the severity of the phenotype varies amongst patients that all have the same genotype. Polygenic traits means that multiple genes contribute to the disorder. Individuals can have gene-gene interactions. We know that genes work in pathways, so depending on an individual's overall genotype, right, as that varies from one family member to another, they may have different phenotypes. And lastly, the environment certainly interacts with genes. So the consequence of these types of complicated inheritance patterns is that it's much harder to identify the disease genes. However, we have made a lot of progress, and many of these genes are known. However, the pedigrees that we'll look at for monogenic disorders are not as useful. Here would be an example. This would be a typical autosomal recessive pedigree. You can see individuals that are labeled, unaffected, big A, or big A, little a, if they're heterozygous. And in those individuals, the symbols, the squares for the males and circles for females would be half filled in if they were heterozygous carriers of the disease allele, which in this case is little a. But the idea here is that in a complex disease, when you look at generation 3, the bottom row, you'll see that only 2 of the 7 individuals are going to have the little a, so it's relatively rare, and only one of them, the circled person in red on the left, it may be the only one who actually shows the phenotype. And again, this is because of potentially different lifestyles, different interactions with the environment, or just other gene-gene interactions that the second person who's little a doesn't have. And we'll talk more about pedigrees in our next session. So how can the environment interact with the genome? It is at a molecular level, and there are some really interesting examples in Table 1 in the complex disease chapter of that citable online book that I mentioned earlier. The link is shown there in green. 2 examples are skin cancer, where individuals that have mutations in the MC1R gene, the melanocortin-1 receptor, are going to be at risk for developing skin cancer, and it's only upon exposure to UV that skin cancer actually occurs. Similarly, acute alcohol sensitivity, individuals with mutations in ADH1C, which is alcohol dehydrogenase deficiency, those individuals are going to be predisposed or at risk for this phenotype manifesting itself when they drink alcohol. Okay, so those are just 2 examples of how the environment can interact with the genome. So for these complex diseases, I said that pedigrees don't work well to identify genes. And so what does work, and how have we been successful so far? The answer is often genome-wide association studies. These are abbreviated GWAs, and as defined by the National Human Genome Research Institute, it's an approach that involves looking for markers across the genomes of many individuals. And what you're doing is you're looking for genetic variations that are associated with the disease. In this case, you're not necessarily looking first for the gene itself, but just for correlations, right? How do the genomes, what do they have in similar with one another with individuals who have a particular disease? So this has been an effective approach. It is time-consuming and expensive, but we have been able to identify genetic variations that contribute to these common complex diseases like asthma, cancer, diabetes, heart disease, and mental illness. And as of 2011, 400 regions of the genome were associated with 70 different diseases or complex traits. So it certainly has been effective, although there has been some controversy over the years because it's been a little bit slower going than people had hoped. Okay, so Once you have selected a potential topic, what you want to do is go to OMIM. So everybody should do this. Once you have a topic in mind, you want to go to this website that's at the top of this slide. And what OMIM is, it's called Online Mendelian Inheritance in Man. It has many, many overviews for all the known Mendelian disorders, over 12,000 different genes, and it's basically an online textbook of sorts that includes lots of references. You will need to find actual research papers, a combination of primary research articles and reviews for your research paper. You will not be able to use any online sources. This is clearly laid out in the guidelines for the research paper. So OMIM is one of the possible places where you can find those research articles. Okay, so I'm going to be going through the next several weeks with you, showing you how to think about the questions that you need to address in your research paper, and I will try to model how to do that using the disease cystic fibrosis. So this topic is off limits for anybody else in class to use because I've already chosen it for my topic. If you type in cystic fibrosis in the search bar at the top of the OMIM website, this is what you'll see. And interestingly, you'll see that I've circled in red the disease itself. It is going to have the number symbol, which you can see here. I've put a little number sign here. So you can see that next to the number 21970, that is preceded by a number sign. Okay, so that's the disease itself. In addition, when we do that search, we also get the gene that's associated with the disease, and that will be indicated with an asterisk. So I've boxed this entry number 6. It has an asterisk, 602421. Both of these are going to be useful links because in your research paper, you will be describing the disease, but you'll also be describing a particular gene that you select that's associated with the disease. Remember, with those complex diseases, there may be multiple genes from which you can choose. For cystic fibrosis, there's only one, and it's the CFTR gene. Okay, so let's see what we can learn when we click on these links. Oh, first, I forgot, I pasted in here just an overview, excerpted from the guidelines for the research paper. Just to remind you that you're going to be introducing the disease in this first section, common symptoms, the course of the disease, if it progresses with time, does it get worse as people live with the disease longer? What's the inheritance pattern and what's the frequency in the population? You'll then move on to talking about diagnosis of the disease, and then treatment. In the latter sections 4 and 5, you'll talk more about genes associated with the disease. So, section 4, how was the gene identified? How was it mapped to a particular chromosome? And how does the gene function? So, when we click on the protein encoded by the gene, you'll see on the far left of this page that there's a table of contents. And you can see that the text includes information on how the gene was cloned, how the gene is expressed, the mapping of the gene to a chromosome, how the gene functions, as well as animal studies, which will be also important for us to understand how the gene functions. If you see all of these chapters essentially in the text, that tells you that there's a lot of information known about this topic and it will work for this research paper. If, on the other hand, you don't see these key headings or subheadings, the cloning, the mapping, the gene function, then that would be an indicator the topic may not work very well, and I can help you with that. If you click on the disease, which was, remember, the link that had the number sign, this is what you might see. You'll see clinical features, inheritance, which again is something that we're looking for, pathogenesis, diagnosis. Those are also going to be useful sections where we'll be able to click on those and see links to specific research articles that have information on that topic for this disease. So this is an example where I clicked on one of the mapping links, and what you would see is that for cystic fibrosis, Rorton published in 1989, where that research group was able to map the CFTR gene to chromosome 7. And then it tells you there's yet another link about mapping the cystic fibrosis gene. So the Rorden paper is actually one that we're going to be reading and discussing next week, and we'll see some of the methods they use to map the gene to a chromosome. Okay, so at the bottom of this slide, I'm just reminding you that this is a great tool to find these primary research articles, and just a reminder that you cannot cite OMIM itself. Okay, so to wrap up for today, your goals before Thursday are to pick your research paper topic carefully and get it approved and email me at my email address that's on this slide before our usual class time, 11 a.m. on Thursday, March 19th. Make sure you're choosing a specific form of genetic disease. For example, there's 9 forms of muscular dystrophy. So you need to pick one form, and eventually you'll need to choose one gene that's implicated in causing the disease. The second thing you need to do before class on Thursday is answer the pedigree reading guide questions that are posted as an assignment in Blackboard, and you'll upload your answers as a Word document in Blackboard. This assignment will be replacing the reading quiz, and we will probably take this basic approach, not having reading quizzes while we are not meeting face-to-face. Okay, so in close, I just want everyone to feel free to contact me with any questions about anything we've talked about, and use those discussion boards within your teams to talk with one another and help each other out. And I'm looking forward to seeing what topics you all pick and learning a lot from you as you start drafting these research papers. The first draft is going to be due March 31st, so as soon as you pick your topic, you'll want to start working on it. All right, take care until next time, and that is all.